not least, a presenter of this afternoon, Courtney Harrison. So Courtney is a third-year master student with Dr. Thomas Locke. She is interested in disease ecology. With a bachelor's degree in animal and nutrition science from West Virginia University, Courtney today will give us a presentation on field and laboratory-based experiments, provide evidence that canobacteria infections may be a factor in poor lake whitefish recruitment in the Great Lakes. All right, so please go ahead, Courtney, when you're ready.
Okay, thank you so much for the introduction. So first, I want to thank my co-authors on this study, Dr. Travis Brendan from the Quantitative Fisheries Center, and Mark Ebner from the Fresh Lake Whitefish Company. I'd also like to thank my colleagues at the Michigan State University Aquatic Animal Health Lab. So today I'm excited to present some research findings that involve an infectious disease to be a possible factor in poor Great Lakes lake whitefish recruitment. And so for those of you who don't know, this is the lake whitefish, Coregonus clupeiformis. It's an important indigenous fish species within the Great Lakes because one, it plays a critical role in cycling energy throughout the Great Lakes food web, but it's also one of the most highly valuable commercial fish species within the 4 upper Great Lakes. But unfortunately, their population numbers have fluctuated substantially. And so what you're looking at here are lake whitefish yields over time. So you can see that they reached an all-time low in the 50s, in part with the introduction of sea lamprey to the Great Lakes. And although their numbers did recover, they declined once again. And over the past decade, we're still seeing these similar trends. And along with that, there have been declines in adult abundance and growth. And so some studies suggest that some contributing factors behind these declines could be from changes in the Great Lakes food web and invasive species, among others. And along with the declines in abundance and growth, we're also seeing declines in early life stage recruitment. And so this current study is centered around recruitment. So before I move on, I just want to make sure we're all on the same page. So what is recruitment? Simply put, it's the introduction of new individuals into a population, which means that eggs need to be successfully laid, fertilized, they need to hatch, and then the fry need to survive to join the adult populations. And so seeing declines in recruitment of young lake whitefish is concerning, especially because we don't know what's causing these declines. And infectious diseases as a role in recruitment hasn't been thoroughly investigated. However, this doesn't mean that we don't know of any infectious diseases infecting Great Lakes lake whitefish. And so several pathogens have been found to infect adult Great Lakes lake whitefish, as you can see here in these studies. And though some of these pathogens were recovered from diseased fish, does the pathogen actually cause disease? This is a huge knowledge gap. And though these pathogens were recovered from adults, what hasn't really been looked at is whether or not these same pathogens are in the reproductive tissues of these fish. And so in the fish health world, there are a multitude of pathogens that can infect the parent and subsequently infect the offspring. So the pathogen uses the reproductive tissue as a sort of vehicle for infection. And again, this is a knowledge gap. So if they are present in the reproductive tissue, does that lead to infection in the eggs? And if the eggs are infected, does that persist to infection in the sac fry? And likewise, would those infections persist in later early life stages? But above all else, what do the effects infectious diseases have on the health and survivability of this host, the lake whitefish? And so last year, if you can remember, I presented my thesis concept involving a larger study looking at an array of pathogens that can be vertically transmitted from infected parent to their offspring. And so to get a better understanding of the pathogens in these spawning fish, I began by sampling spawning adult lake whitefish at multiple locations that you can see here. So I selected sampling sites in both Lake Michigan and Huron that reflect both good and poor recruitment. And so I already introduced the term recruitment, but what's the difference between good and poor recruitment? So in these good recruitment sites, you're going to see high persistent biomass, whereas in poor recruitment sites, you're going to see declining biomass and less overall abundance. And also in these poor recruitment sites, there's evidence of seeing fewer age 0 fish. And so with that, I selected 60 fish from each site, totaling 120 fish from each lake. And in the process of doing that, I found some pretty serious fish pathogens in these adults, including Flavobacterium psychrophyllum, Rhenibacterium salmoninarum, Motile aeromonas, and Carnobacterium. Before the remainder of the time today, I'm going to focus on this one in particular, Carnobacterium. So what's Carnobacterium? It's a gram-negative bacterium in the family Carnobacteriaceae. It's the causative agent of pseudokidney disease, and in other salmonids, it has a penchant for reproductive tissues and is known to cause post-spawning mortality. And so with that, the overarching hypothesis is formed. Carnobacterium infections are negatively affecting Great Lakes lake whitefish recruitment. And to address this hypothesis, I have a set of main objectives, with one being to assess the prevalence of Carnobacterium in adult and juvenile lake whitefish, and two, to determine the pathogenicity of Carnobacterium in juvenile lake whitefish under controlled laboratory settings. And so to begin, I'm going to start with this first objective with an emphasis on the adults. So during the course of clinical exams and analyses on the 480 fish that I collected from both good and poor recruitment sites in Lake Michigan and Huron, I noticed some gross signs of disease, including this one here. You can see some focal hemorrhaging, focal intraocular hemorrhaging. And when we opened up these fish, I also noticed some gross internal signs of disease. So a subset of fish had some severe hyperemia of the swim bladder vasculature, which just means the engorgement of the blood vessels with blood in the swim bladder. And when we opened up these swim bladders, I saw this mucoid turbid fluid. And then in some instances, the fluid was more severe in other swim bladders. Like the one you're seeing here, you can see fluid is filling up almost half the space of the swim bladder. And among others, I noted too some internal signs of congested kidney in these adult fish. And in all cases, I took bacterial cultures from this fluid within the swim bladder and from kidney cultures, and I recovered this bacterium with this morphology. And so remember, a main goal of this study is focused on recruitment. So I definitely wanted to look at the reproductive tissues. So I looked at the testes and ovaries of adult spawning fish, and I took bacterial cultures of each. And surprisingly, I found the same bacterium within the reproductive tissues. So now a major goal moving forward is, what is the identity of this bacterium? So once I verified culture purity, I performed typical phenotypic and biochemical characterization tests on 14 isolates collected from 13 adult fish. And in all cases, this bacterium was gram-positive, oxidase negative, and catalase negative. And so I won't go through the rest of the tests with you today, but I was able to presumptively identify this bacterium to belonging in the genus Carnobacterium. And to get a confirmatory identification, I performed PCR analysis on the 16S rRNA gene, and I got these bands you're seeing here. Then I purified the amplicon and performed sequence analysis. And from this analysis, I placed the bacterium I recovered into the genus Carnobacterium. And so for an even more definitive identification, I performed phylogenetic analysis. All right, so what you're looking at are all of my isolates compared to all the described Carnobacterium species. And so I color-coded mine to depict the year the isolates were recovered, so yellow being 2018 and green being 2019. And then the shape depicts the location that they were recovered from. So you can see I have isolates from both Lake Michigan and Huron. So a big takeaway from this is that compared to all described species of Carnobacterium, all of my isolates fall into a robustly supported clade that contains the type strain Carnobacterium ultramaticum, meaning that all of my isolates are indeed Carnobacterium ultramaticum. And so now I'm just going to take you over the prevalence of Carnobacterium infection from both lakes. So I have the prevalence on the y-axis with the location on the x-axis. I'm also, again, showing both good in green and poor in red recruitment shown by these stars here. And so interestingly, in Lake Michigan, there's a higher prevalence of Carnobacterium infections in the poor recruitment sites during both years. And in 2018, in Lake Huron, there was a similar story. Again, the prevalence being twice as high in the poor recruitment site when compared to the good recruitment site. And then in 2019, the prevalence was the same. And so to wrap everything up about what I found in the adults, I found Carnobacterium in fish showing signs of systemic disease. And for the first time, I recovered the bacterium in the reproductive tissues of spawning fish. So this means this could also potentially infect their offspring. I also found higher prevalence of Carnobacterium infection in poor sites overall. So now that we have a better understanding of Carnobacterium infection in the adults, we obviously want to look at infection prevalence in wild juveniles. But unfortunately, 2020 happened, and with the onset of COVID, I did not have a field season. So today I don't have that data to present to you, but hopefully I am going out again this year to get at this important aspect of the study. But despite this, we still need to address objective number 2. So what effects does this bacterium have on the host? So we know that the bacterium can infect the host, but does it cause disease? So in collaboration with the Little Traverse Bay bands of Odawa Indians, I received juvenile lake whitefish. So eggs were artificially fertilized from wild broodstock originating from Elk Rapids in Lake Michigan. Eggs were disinfected with erythromycin and iodophor, both on site and once again before entering the hatchery facility. And so once the eggs hatched, they stayed at the hatchery until about 3 months of age. And then they were transferred live to the Michigan State University Research Containment Facility, where they were held under quarantine conditions in UV treated water. And so as I continued to maintain these fish, once again, 2020 happened, which delayed the start of my experimental challenges. And so in the process, my fish continued to grow. Unfortunately, in October of 2020, I started to see the development of disease signs in my fish. So I started seeing linear ocular hemorrhaging, like in the picture you're seeing here with this fish. In other cases, I saw some severe exophthalmia. And then I started seeing more severe ocular hemorrhaging. And then as time passed, I started noticing more chronic signs of disease. Like in this fish here, we still see some exophthalmia, but now we're seeing some lenticular and corneal opacity. And in this fish, we're still seeing that lenticular opacity, but now we're seeing corneal perforation, which just means that the lens is perforated through the cornea. And when I opened some of these fish, I noticed some visceral edema. So collectively, this indicated that my fish were showing signs of a systemic disease. And so my level of concern was growing because remember, I was planning to use these fish for experimental challenges. So I needed to know what was causing these infections. And so I decided to investigate further, and I took a similar approach as I did with the adults. So I took kidney, spleen, and heart tissue from these fish, and I inoculated them onto 3 different cell lines. And in all cases, there was no cytopathic effect on any cell lines, so I could rule out that this wasn't a virus. So then I started taking bacteriological cultures of these fish, which included the kidney. And then again, remember, they were showing severe signs of exophthalmia, so I also included bacterial cultures of the eye and the brain. And in some instances, I got these intense bacterial infections. Again, all coming from the kidney, the eye, and the brain of these juvenile fish. And so again, I used the same approach as I did with adults, and I performed bacterial analyses. And later, I came to a presumptive identification of this bacterium belonging in the genus Carnobacterium. And once again, to get a confirmatory identification, I ran the same PCR analysis on 11 isolates from 9 juveniles. And again, sequence analysis showed that the bacterium belonged to the genus Carnobacterium. And so now you're seeing another dendrogram, but this time I added the recovered isolates from the disease outbreak in the lab, shown here by the purple circle. And then I also added all the isolates I recovered from adult lake whitefish. And once again, all of my isolates fell into that robustly supported clade containing Carnobacterium ultramaticum. And so while trying to determine the cause of this, disease outbreak, unfortunately, antibiotics was not an option because remember, these fish were destined for experiments with bacteria. So in the meantime, I made some adjustments. So I modified rearing methods. I also took note of hygiene of the fish, and I used non-chemical treatments like salt baths. And at the point of the outbreak resolution, despite my best efforts, disease outbreak led to a cumulative mortality of just under 25% and just under 35% in both of my tanks. And so at the end of the day, and even though I was concerned because again, this was unplanned, and although this was unplanned, and they were showing signs of this infection, Corynebacterium was one of the target pathogens that I wanted to study in the first place. So I thought, when life hands you lemons, or in this case, 2020 hands you lemons, you have to learn to make lemonade. Because after all, the result of all of this was that this is the first Corynebacterium disease outbreak in Great Lakes Lake whitefish under laboratory conditions. And so one possibility was that maybe this bacterium persisted in juveniles and led to systemic disease and mortality. And so to summarize everything, I found Corynebacterium in diseased adult lake whitefish. I also found Corynebacterium within the reproductive tissues of spawning adult lake whitefish. And now we know that Corynebacterium negatively affects the health of juvenile lake whitefish. So collectively, findings uncover that Corynebacterium ultramaticum might be a possible factor in poor recruitment of lake whitefish to the Great Lakes. And so at this time, I'd like to acknowledge the Great Lakes Fishery Trust for funding this project, the fishing boat captains for letting me come out on their boats with them to collect adult fish, the MDNR Fisheries Division, the Little Traverse Bay Bands of Odawa Indians, my friends for helping me collect fish, all of my colleagues at the Michigan State University Aquatic Animal Health Laboratory, and my advisor, Dr. Thomas Locke. And I'd be happy to take any questions at this time.
Thanks very much, Courtney, for the presentations. Great job. Any questions for Courtney? Oh, I think Tyler raised his hand first, so I'll go with Tyler first.
Hey, Courtney, great talk. I'm curious if Corynebacterium is something that shows up as a primary infection, or if it's something that can be more common in already stressed individuals.
So we know that Corynebacterium tends to occur in more stressed individuals, although the bacterium itself is a ubiquitous bacterium. It's found within the environment, within meat and poultry too. But again, like we found these fish, the main point is that we found this bacterium in spawning phase, like whitefish. So of course there's going to be stress coming along with spawning. So that's something important. So it may, it's more likely to react. So whenever they're stressed.
Nice.
Yeah. Mm-hmm.
Next we got Gary here.
That was a nice talk, Courtney. I appreciate you working on that fine species that we're having plenty of issues on right now.
Thank you. Yeah.
So my question for you, could you remind me what the mode of mortality is with this particular bacteria? What is it affecting and how is it actually causing the mortalities?
How is it causing the mortalities?
Yeah. What, for example, what organs is it affecting, and is it causing, for example, mass organ failure? I mean, how do you think it kills the fish, other than just an infection?
So there's still a lot of work to be done with that. Again, these experiments were planned, and unfortunately, it just so happened that they were infected with the bacterium, and we know that it causes pseudokidney disease. So the kidney would be one of the primary organs that it would affect. However, I was finding these really intense infections in also the kidney, but also within the eye and the brain.
Mm-hmm.
So there is some, potential for this bacterium to really cause some damage in those tissues.
Looking forward to seeing more. Thanks.
Thank you.
And we got another question from the chat box from Mary. To what extent do you think the prevalence of a particular bacteria varies, especially in Lake Michigan, versus the vulnerability of the fish varies especially?
I'm not sure I understand this.
Mary, would you like to elaborate your question a little bit?
Sorry, I was multitasking. Hi. Yeah, well, just if there are areas of poor recruitment that correlate with more prevalence of the bacteria, it just got me wondering, is it really the bacteria that's varying spatially, or is it the fish's vulnerability to the bacteria that varies spatially? And in a way, I think all our questions are getting at the same question in different ways. I think my question is similar to Tyler's.
Right. So I definitely think that, you know, there are other aspects that, you know, could cause harm. It obviously would stress the fish. So environmental changes, water temperature, pollution. I mean, there's all sorts of things that could come into effect. So I think, yeah, I do think that it depends on the environment. It also depends on the host too, on whether or not we'll see more of these infections.
Yeah, thank you, great talk. And I think you're making fine lemonade, so well done.
Thank you.
Yeah, thanks. We don't have any questions, but we have one comment from the public in the wild. I would think vision impairment would affect the ability to evade predators. From William.
Yes.
All right. I think this is it. Thanks, Courtney, for the presentation.
Thank you.
And I'm glad that I moderate these 5 outstanding presentations this afternoon. And thanks everybody participation and interaction in this session. Now I'm giving back the host to Emily and Amber for the